Hi, my name is Meredith Hartley, and I'm an assistant professor in the Department of Chemistry. My lab works on the role of lipids in neurological diseases, and we're particularly interested in these diseases that have myelin damage. Myelin is a really critical component of the brain and spinal cord that is a lipid-rich membrane that surrounds axons to protect them and enhance signal transduction. And when myelin is damaged, this leads to a number of different symptoms and is implicated in several diseases. The most common one is multiple sclerosis. And so we're really interested in figuring out how lipids are regulated during the process of myelin damage and myelin repair. In my postdoc, I was in Oregon at Oregon Health and Science University. And there I was working on myelin diseases, but in the context of developing therapeutics for them. And so I actually tested a number of analogs of thyroid hormone called thyromimetics as potential therapeutic small molecules that could actually promote myelin repair. And so we were able to show that some of those were effective, and a few of them are actually going into clinical development for multiple sclerosis. So as I started my own lab at KU, I have always had a longstanding interest in membranes and lipids and how they interact in various disease states. And so it was kind of a natural shift for me to start looking at the lipids in myelin. And in the course of my previous research, I had worked with a really useful animal model of myelin damage and repair that I thought could be a really good platform for us to start looking at how lipids change during these processes. Well, my research group is doing everything, and I don't do anything now. It's kind of an abrupt change from being a postdoc where you are in the lab all the time to being a professor where you don't really have time to be in the lab all the time. The chemistry department has been really supportive, and students have been really enthusiastic, which is great, about joining the lab. And so we've really been able to build quickly and start to get some results. There's a number of different projects going on. We have one project where we're actually using mass spectrometry to measure the lipids at different stages of myelin disease. We have another project where we're using a cellular model to hopefully identify genes that are important and relevant during these pathways. And then our ultimate goal is to kind of synergize these two data sets to hopefully identify pathways that are really critical for myelin damage and repair.